Good afternoon, everyone, and welcome to today's session in our Cellular Conversations webinar series. I'm Alex Sabo, CEO of Millennium Science, who is the proud distributor for 10x Genomics in Australia. We are absolutely delighted, I should say, for the terrific interest in today's session, focusing on long-read DNA sequencing and single-cell analysis, with the focus on quantifying RNA isoform expression. As you know, short-read sequencing has been the default platform to date, but we've actually had a record number of participants register for this session, so it's clear that today's topic has really struck a chord. For those of you that are new to Cellular Conversations, what we feel makes this short webinar series different is that it's not just about showcasing results, it's about going under the hood, so to speak. Our goal is to focus on generalizable insights into experimental design, practical tips and tricks, inevitable challenges that you might face in the lab in doing single-cell analysis, and how to overcome them. So we hope that this can be a forum for real, candid knowledge sharing, the kind of insight that you expect to get from a colleague at the bench next to you. We're going to dive into the topic of the day in just a moment, but to put things in context, I'd like to hand things over briefly to my co-host, Jeff McDermott. Most of you know Jeff. Jeff is a science and technology advisor with 10x Genomics, and Jeff's going to briefly speak about some information about 10x products that are relevant to today's seminar, in case you're inspired by today's talk and are interested in pursuing similar studies. So over to you, Jeff.
Awesome. Thanks, Alex. And I assume you can see my slide okay.
Yes.
Perfect. I'm going to be really quick here. Two quick things. First of all, thank you so much for coming along today. We always appreciate you taking time out of the day to join these webinars. And second thing I want to mention is that in Sefi's webinar, he's obviously going to be covering long-read sequencing for single-cell transcriptomics. Of course, long-read sequencing and single-cell have been paired together for a while now, but I just want to highlight that we just launched a new product that we call Visium HD 3'. And this uses a reverse transcription-based biochemistry to capture spatially resolved whole transcriptome gene expression. So what you're about to see today in terms of long-read sequencing can also be applied spatially if using the Visium HD 3' product. So just wanted to make you aware of that not only can you do this in single cell, but now it's possible to do it all with spatial as well. And with that, I'll hand it back.
Perfect. Thanks, Jeff. So with no further ado, it's an honor and delight to welcome today's speaker, Dr. Sefi Prawer, who is a research fellow in the laboratory of Mike Clark at the University of Melbourne, specializing, as we said, in long-read and single-cell transcriptomics. His work focuses on developing and applying advanced sequencing technologies to unravel the complexity of RNA isoforms in the human brain, particularly in the context of neurodevelopment disorders. In addition to his bench work, Sefi has been a key contributor to the development of informatics tools for the analysis of nanopore sequencing data, including the tools known as NanoCount and the Red Hot Blaze and Flames. In taking on these ambitious challenges, it's noteworthy that Sefi also has a degree in astrophysics. So clearly, whether navigating stars or splice variants, he has a knack for illuminating dark matter. Sefi, we're excited to have you with us. Take it away.
Can everyone hear me okay?
Yes.
Great. Wow, what an introduction, Alex. Thank you for that. No one's brought up my minor in astrophysics for quite a while, so that's pretty cool. All right, so today I wanted to talk about the hidden world of RNA isoforms and the tools and technologies in the lab that we've been using to kind of study these in single-cell resolution. So first of all, what are RNA isoforms? So we know that genes can produce many different RNA products through a process called alternative splicing. These different RNA products are referred to as isoforms. And these isoforms can have different functions, both in development and disease. We know these RNA isoforms play important roles during this developmental and disease processes, and our interest is studying these in a little bit more detail. So the central question in the lab is, how does a single cell develop into complex organs and tissues in our lab, we're focused particularly on the brain. We know that isoforms play important roles in these processes, and if we understand the development of this process in the context of RNA isoforms, we'll get a better understanding of neuropsychiatric disorders and diseases, which is something that we also discuss a lot in the lab. So in the context of the developing brain, we know that nearly all human genes have multiple RNA transcripts. These transcripts have been shown to be involved in key processes during development, including neural differentiation, neural migration, and synapse formation, just to name a few. And this figure that I'm showing on the right is from a review from 2018 by Su et al., showing these key splicing factors that are fundamental during these developmental processes, highlighting the importance of these different isoforms during development. So the question that we're focused on in the lab is, can we use single-cell RNA sequencing to look at the isoforms in different brain cell types and along this developmental process to understand which isoforms are present in which cells and how they affect this developmental process? So we know, especially everyone in this webinar at the moment is aware of the power of short-read single-cell technologies. Just to name a few, we can identify rare cell types and plot developmental trajectories. And most of us in this room have probably used these technologies to do exactly this. But these technologies are limited because they only profile gene-level expression due to the nature of the technology. And there's no information on isoforms. So to expand on that in a little bit more detail, the difference between short and long read sequencing, there are a few fundamental limitations when it comes to short read sequencing. Primarily, there's a process of fragmentation, and this fragmentation means that we generate short reads, and these short reads in the context of 3' or 5' gene expression assays from 10x are usually biased to the 3' or 5' end of the gene, meaning there's no long-range connectivity information. We don't know which exons are connected to which exons, generally speaking, and there's this bias at the 3' or 5' end. But for long-read technologies, there's no fragmentation step. And this means we have the ability to get full-length, complete RNAs that we can map back to the genome. And this enables us to look at both gene expression, isoform expression, and also identify novel isoforms that previously haven't been characterized. And I show a quick example here in this red box down below. So this has been the central focus of my work in the Clark lab for the last few years. And in order to make this a possibility, we've combined long-read and single-cell technologies together. First, we published the Flames package, which is a complete suite of tools and pipelines to analyze long-read single-cell data. And that was published in 2021. And then during the end of my PhD, we published Blaze, which is a tool for cell barcode identification from only long-read data. Since then, we've made some advances to the Flames pipeline, and it now involves lots of different options, and it's been enhanced in a lot of different ways since it was published in 2021. This includes the option to only use long reads and no longer the requirement to use short reads if you don't want. However, the option is still there to use both technologies at the same time, and these technologies can supplement one another. And additional technologies that will improve identification and quantification of both genes and isoforms. So generally, the tool's faster, only requires long reads, and supports single cell and spatial technologies as well. It's available at Bioconductor if anyone is interested in giving it a go, if they have some long read data that they wish to process. So in the lab, we're really interested in this process of brain development. And one of the key tools that we use are cortical organoids to model this process. We use the ALOTA protocol, which is a very famous protocol published in Nature in 2019. And here are some of the organoids from this protocol in a bioreactor. at different time points along development. And they produce similar cell types that we would see in the fetal brain, and they're really robust for studying this brain developmental process. So one of our experiments has been to take these organoids from different time points and apply the 3' 10x assay on 8 organoids, 2 from 1 month, 3 from 3 month, and 3 from 6 month. And apply the 3' 10x assay. And then instead of doing short read sequencing as conventionally done, we supplement in a long read protocol where we sequence the cDNA from these organoids using nanopore sequencing, and then process this information with our packages. This enables us to have a look at isoform-specific expression in individual cell types. So just like short read single cell sequencing, with long read technologies, we can identify cell types, we can identify rare populations as well, and we can plot some UMaps based on gene expression and isoform level expression. And here are some UMaps shown below. You can see that we can identify a wide range of cell types that we would expect to see along the developmental process. But for those who aren't experts in brain cell types, the general cell types that we expect to see are these radial glial cells, which are a type of progenitor cell, some early neurons at 1 month, then some excitatory neurons that start to develop at 3 months, and some more mature cell types like astrocytes, excitatory neurons, and inhibitory or interneurons. So we can see all the range of these cell types across development. Similarly, we can also identify marker genes, but not just marker genes, we can also identify marker isoforms. So you can see that there's a wide range of marker isoforms that kind of are unique to these different clusters. And what's really interesting about marker isoforms specifically is that when you have a look at these marker isoforms, the vast majority of them are only found in one particular cell type, meaning that these marker isoforms are specific markers that kind of are very useful at identifying which cell types are present. So we can do this at the gene level, which most of you have probably done before, but we can also do this at the isoform level as well, meaning that we have both gene and isoform level information within our data. Similarly, we can plot these marker genes on a volcano plot. And here I've just listed a couple of known markers for inhibitory neurons. So we can see the downregulation of markers that are not associated with GABAergic or interneuron development, while the 2 main markers, GAD1 and GAD2, which have to do with the GABAergic synapse, are the largest hits that we can see on the right-hand side over here in a volcano plot. This is exactly what we would expect to see at the gene level. But in addition, we can also do this at the isoform level now as well, adding this extra layer of information. So here I've just listed some of the GAD1 isoforms, and we can see they're also upregulated in inhibitory neurons. And we get some extra information showing which isoforms here are highly expressed when it comes to the inhibitory neuron cluster. We can take these isoforms and do Go enrichment analysis, and hopefully you can see GABAergic related processes and neural developmental processes associated with both these marker genes and marker isoforms. So we can also have a look at the isoform landscape generally. So which isoforms we find. So we sequenced about 700 million long reads on the Promethean flow cell, so 1 flow cell per sample. And from this data, we generated more than 170,000 unique isoforms, close to which 10,000 of which hadn't been identified before. So totally novel, not present in the reference. We can categorize these isoforms as well based on their structural categories. So the blue bar represents isoforms that match the reference, and these other categories represent novel isoforms that fall into different novel categories. Either they have a novel junction or a novel exon, or maybe they have known exons that are kind of stitched together in a different way that hasn't previously been identified. And this is really a powerful application for long-read technologies that were able to identify isoforms and structures that we previously couldn't identify with short-read technologies. So just to highlight some interesting examples of this, here's a list of a few of our genes that have the most isoforms, and these are all isoforms expressed in this data set. And we can see some of these genes express more than 100 isoforms. So just want to highlight the complexity of the data here. What we're actually able to identify means that we can really dive under the hood and look at a lot more than just gene level expression. To summarize this in a nice bar graph, we can see that many of the genes that we sequence have a single isoform, and this is primarily due to the amount of sequencing depth that that we have. But a lot of the genes that we find have multiple isoforms, as kind of showcased by by this graph over here. So let me just highlight some interesting examples just to showcase what additional information we get from long-read technologies. So here is an isoviz plot. Isoviz is a software that we developed in the lab to visualize isoform structure. So I'll show a few of these plots. So just to orient everyone, this is the gene TBM1M6. Here is its protein domain, and the boxes show exons and the lines show introns. At the top is the canonical isoform, so the isoform that is used most dominantly, I guess. And here is the isoform plus a new isoform underneath, which is labeled with this bamboo ID. And this is because it's an isoform that's novel. So it's not found in our reference. And I've highlighted in red here the part that is novel, where we have a new splice site that occurs at the 3' end of this gene. So if we look at gene level expression, we can see there's expression across all clusters, but it's localized to this cluster over here, which is an astrocyte cluster at the top. But we can also now zoom in and have a look at isoform specific expression. And hopefully what you can see from these feature plots is that we see enrichment of this novel isoform in astrocyte specific cells. And this is something that you will not be able to see at the gene level. You need to kind of zoom in a little bit and look at the isoform level to see this novel expression, which is localized to astrocytes. Another example of what we call isoform switching during the process of brain development is this example of GPM6A. So we know that this is enriched in astrocytes and it is involved in brain signaling and transduction. And at the top over here, again, I'm showing an isoviz plot with now two different known isoforms, one long one and one short one. So this is the expression at the gene level. It looks kind of uninteresting, I guess. It looks like there's ubiquitous expression across all these different clusters. Here I've just labeled a couple of the different broad categories of clusters. So the astrocytes, the inhibitory neurons, and the excitatory neurons. And at the gene level, there's no significant difference in expression. But again, if we zoom in and have a look at the isoform-specific expression, we actually see something that's quite different. You can see this long version of the isoform, this one over here, is primarily localized again to astrocytes, and it doesn't really show expression in any of the other cell types. Conversely, the shorter isoform is predominantly expressed in these two bottom clusters, so inhibitory neurons and excitatory neurons. And we can quantify this, and we can actually see that there is a significant difference in isoform usage across different cell populations. So this is an example that highlights that even though a gene can be expressed ubiquitously or expressed in a particular context, the isoform level expression may give you a different kind of picture, may give you a different understanding of what the cells are actually doing. These two isoforms actually have different open reading frames. So it's suggested that they probably have different protein functions, which is, again, something that's quite interesting because at the gene level, you wouldn't see this. You'd only see this when you zoom into the isoform level. So GPM6A is actually a really complicated gene. I'm showing just a subset of all the isoforms that we find in our organoid data. Before I was just showing two, and here are a couple more. We can also categorize isoform usage based on the canonical versus non-canonical expression of isoforms. So that is to say, we usually expect isoform expression to be localized to the canonical isoform, the dominant isoform. But if we plot the expression based on these different cell types, based on the usage of canonical versus non-canonical isoforms, again, we can see a really different picture. We can see that some populations like the astroglia have a really different profile when it comes to interneurons or even outer radial glial cells. And this is just one example of many to highlight that the usage of isoforms can really vary across different tissues and different cellular contexts. We can do this a little bit more specifically. We can plot the isoform usage again of GPM6A based on the top expressed isoforms. And here there are actually 3 dominant isoforms expressed in astroglia, these 3 different colors. And hopefully you can see the vast variation between isoform usages across different cells in this organoid sample. So we can look at isoform switching events. We can also do something, we can also look at fusion transcripts using long-read single-cell data. So this is an example of the Xenopus court locus, which is a naturally occurring known locus where we have transcription read-through. And in this context, we're actually able to identify a novel isoform at this locus that involves both the court and the Xenopus gene, and it's a fusion of these 2 genes next to each other that we're able to identify. And this is also an isoform that is enriched in progenitor cells in our early population. So this is something that you wouldn't be able to see with short reads because you would not get this long-range connectivity. But with this long-read data, not only can we identify fusion transcripts, but we can identify novel fusion transcripts enriched in different populations. Again, there are things that we can do with long reads that we can't do with short reads. One of them is identifying novel exons. Here is just one example of a novel exon that we find, which is again enriched in a particular cellular context. In this case, it's enriched in this cell type over here. Here is the novel exon that's not previously found in our GenCode annotation. And it actually introduces an early stop codon, meaning that this novel isoform is likely non-coding. And we find 17,000 more or less novel exons in this data set. And there's obviously potential to find a lot more. We can also identify microexons. So these are exons that are smaller than 50 base pairs. And we can have a look at the enrichment of these isoforms in our different cell contexts. And we can see in this case, excitatory neurons seem to have a higher proportion of microexon enrichment than other cell types, which is something that is really interesting to see and might suggest some type of function for these microexons during this developmental process. So that's just a snapshot of some of the things that we can do with long-read single cell that we can't do with short reads. And in order to make this technology more usable and more accessible to the general audience, for those who are interested in it, we've developed a long-read single cell data analysis tutorial. So that steps through the process of generating your count data all the way to analyzing isoforms and generating some of the figures and plots that I've shown so far. For those that don't like coding so much, we've also developed a Shiny app, and this is an app that just takes in a Seurat object and your GTF file and can plot some really simple plots, including isoform structure plots at the top over here and pseudobulk expression plots of your gene or isoforms of interest. So hopefully I've shown everyone today that long-read single cell has this amazing capacity to explore single cells at this extra resolution, where we can explore the landscape in a single cell context that we just couldn't do with short-read data. We can identify cell types, developmental trajectories, and look at gene expression and isoform level expression. And we can identify known novel exons, fusion transcripts, microexons, and everything else that we're kind of limited by with short reads. What's really fantastic about this method is that it's directly compatible with the 10x platform. If you have 10x cDNA, it's something that can be directly applied to this long read method. So it's compatible with short read technologies, and it's also compatible with obviously long read technologies as well. And we've developed tutorials and Shiny applications to make the uptake more accessible. So with that, there are plenty of people to thank, particularly members of the Clark Lab and funding bodies, and obviously Kat and Paul and everyone at 10X. So thank you very much, and I guess I'll open up to questions.
All right. Terrific talk, Sefi, and short and sweet, but right to the point. Fantastic. Just want to remind the audience, there are already a number of questions that have come in the chat. So you have two options. You can either put something up there in the chat, or if you want to just raise your hand, you have the option to just dialogue directly with Sefi and raise a question directly. So, but why don't we dive right into the question? So first of all, from Walter, curious what method you use for the differential gene transcript expression, if you're happy to talk about that.
So there are a couple of different methods for differential gene expression. The simplest that we're using is the find markers and find all markers function from Seurat. But we've also, which I haven't shown here, is done some pseudobulk level expression as well, both at the gene and isoform level. And the tools that we're using are EdgeR and DSeq, so pretty standard tools, yeah.
Okay, terrific. What is the read depth that you're seeing comparable to short read data sets?
Oh, that's a really great question. There's actually a PhD student working on this exact question about kind of benchmarking both the technologies and having a look to see what type of read depth we need to kind of maximize our discovery. Generally speaking, I would say that the main limitation with long read single cell is just the number of cells you can profile for the sequencing depth, because you need to do more sequencing when you're having a look at isoform level expression, because your counts are split across more features. So that's the main limitation. If you're interested in fewer cells, so you don't need as many cells to answer your biological question, I think long reads really is excellent there, because you get your gene level counts as well, which are very, very comparable to short read expression.
Terrific. That was a question from Philip West, by the way. So another question from Therese Johansson, and I think you may have touched on this already, but which tool did you use for the differential expression analysis?
Yeah, so the differential expression was the Seurat find markers functions, which I showed. But again, yeah, there were different. There are many different options here to look at gene and isoform differential expression. I think the pseudobulk methods are usually a little bit more robust, generally speaking. But I think everyone in the field kind of knows that there's so many different types of differential expression comparisons that you can make.
Terrific. Joanna Puteril is asking, are you picking up any partly processed mRNAs?
Yeah, that's a really interesting question. It's definitely the case that we see what we think are partially spliced transcripts. We have some data sets in the lab where we're looking at single nuclear data, and we actually see the proportion of reads that contain an intron to be much, much higher. So we think it's based on the technology and the context, but we definitely see what we think are partially processed or not completely spliced transcripts. And that is something that can be a bit tricky, because it's hard to know if that is a complete transcript that just has a retained intron, or if it's something that just hasn't fully been processed yet.
Yeah. If I can just ask one question as an aside myself, just struck by the fact that in some cases, as you said, you have very large number of different isoforms, upwards of 100 and so on. What are your thoughts about the role of such a wide variation of different isoforms? Are most of these non-functional, or do they have specific functions in different cell types and so on?
Yeah, it's a really, I guess it's a critical question when it comes to just studying isoforms generally. There's such a diversity of isoforms, even for what we think are simple genes, like that one of GPM6A that I showed before. There are predominantly 2 or 3 dominant isoforms, but there are still so many isoforms that we find in the data set. And we're not exactly sure what the isoforms that have more lowly expressed, what their functions might be. And that's something that we are focused on in the lab, modifying these isoforms, increasing the proportion of the usage of one isoform over the other, and implementing that in different stem cell models and seeing if we can see some type of difference when we change the proportion of usage across different cells. So yeah, I guess it's a tricky question to answer, and it's going to be gene specific. But the diversity of isoforms really does make it a tricky thing to study.
Great. Question from Martin Smith. I presume your UMAPs are generated with gene level quantification. What happens if you use isoform level counts for this?
Yeah, that's an awesome question. So the UMAPs I showed were gene level counts. We can do exactly the same thing using isoform level counts, and we see the concordance for the UMAPs to be very similar. But generally speaking, the isoform counts are more dispersed and there are more features, meaning that the UMAPs are usually a little bit more noisy just because the data is a little bit more noisy. But we do see some cells that kind of cluster based on specific isoforms that don't cluster when you're just looking at gene level expression. So there is some difference between those UMAPs. Generally, the concordance is high though.
Right. Question from Adriana Sana. Amazing work, and thanks for sharing the Shiny app. Is that available also for short read data analysis?
Yes, it would be. It just takes in a Seurat object and will basically plot some feature plots and some structures. If you don't provide a GTF, that's based on I guess you could provide a GTF as well and plot that information, but you won't get any expression at the isoform level. So those heat maps and feature plots at the isoform level just won't be generated. But yeah, you can definitely use it for any Seurat object, any GTF.
Sakini Wirawardina says, "Hi Sefi, thanks for the great talk. Could you please share how you identified the isoform markers?"
Isoform markers being I assume by that you mean the markers that we use with the find all markers function. Those are differentially expressed markers when you compare one cell population against all other cell populations. And we're pretty stringent when it comes to which isoforms we test for comparison. So they need to have at least 5 counts across 10% of cells, 10% of cells within a cluster, because we do see that kind of counts can be dispersed across multiple isoforms of a similar structure. So just to kind of clarify, if you have a read that can be ambiguously mapped to multiple isoforms that have a very similar structure, that read, the count might be distributed over multiple isoforms. So in order to take care of that, we're pretty stringent with which isoforms we actually include in our final Seurat object and our comparisons.
Okay, and I think our final question again from Philip West. Does Flames pick up on all alternative splicing event types? I know some tools have their blind spots, e.g. RMATs doesn't detect intron retention events.
Yeah, that's a really good question. So Flames is a package that's we try to make it as modular as possible. And that means you can use basically any tool that is designed for isoform discovery. So you can supplement the tool, you can supplement any tool into the pipeline. You just need to provide a GTF. The default functions within Flames use Bamboo, and Bamboo is not biased to any particular structural category. So it finds retained introns and categories like that. But if you're not happy with the Bamboo output after discovering your isoforms within your data set, you can supplement in any other technology. The only thing you'd require are your BAM files, and then you can generate a GTF and continue on with the Flames pipeline for quantification.
Okay, thanks. I think that's been all the questions we have from the audience. Sefi, I want to thank you directly for taking such a complex subject and making it, very, very understandable for the audience, and offering so many practical tips for getting into applying long read sequencing in single cell analysis. I assume, Sefi, that you're welcome. You welcome the opportunity to provide advice or to have discussions about potential collaboration with other researchers and so on. So good to see you recognize that. If anybody wants to reach out directly to Sefi, please do so. And also just want to say, for the Cellular Conversations webinar series, if any of you in the audience have suggestions for topics that you'd like us to cover here, please reach out to us directly. You can reach out to Jeff McDermott or any of the Millennium Science staff members. So we'd certainly welcome any suggestions for topics that you would like to see highlighted here. So, but thanks again, Sefi, for a tremendous talk. It's been great to see. I think you hit the nail on the head with this topic, and very informative what you've covered here. Thank you.
Thanks, Alex. Thanks, everyone.